we'll snap on it and see if it's more interest. I'll settle. Come on in, video.
Coming in. Too close?
Yeah, and I think...
No, I'm just slowing down. Still at a little forward way. There you go.
Yeah, so it's definitely in the family Euplectellidae, and I think this is also a Dictyalis, the genus. So yeah, we did get the image of those earlier.
Okay.
There's...
You don't need to take a long look then. Just
And then there's a cup coral there to our left. Those are scleractinian corals. So those are actually the closest relatives that you see at these depths to the shallow water corals that you would see here.
There. Video.
So in shallow waters, the corals that form the reef, that's typically scleractinian corals, and they're cemented together by crustous coralline algae. Those corals that you see in shallow waters, they will have photosynthetic algae living inside their tissues,
Yeah.
and also giving the corals their colors. In the deep, of course, here we are far deep. They do not have these symbiotic algae growing in them, so you have other.
Can snap in.
And so that's going to be another one of these farreid sponges.
Go as far as you like.
So yeah, as I was saying, so in the shallow water reefs you have scleractinian corals, being the dominant corals that form the reefs, and here in the deep,
we're about halfway through the move. The ship is about halfway.
Other groups are more abundant. Several gorgonian and octocorals, black corals, pink corals. There's other groups that are more dominant here in the deep sea.
Okay.
Pull wide.
Thank you.
Looks like the slope is moving more and more northerly.
Is it?
Yeah. Facing 010, and it's a little to the port side.
Okay.
Another 5-10 degrees.
Yeah, we don't want to lose it, so we'll chase it. I leave it up to you.
So I just updated our positions here.
Just stay in your view and chase.
And flater mouse, and we've covered a little over 300 meters now in horizontal and distance, and we've climbed just around 200 meters vertically. We still have
let me just get around a different angle on it.
About 300 meters of vertical relief to climb to the top of the ridge. Now we do have an extended dive, so we do have about 5.5 hours left.
Great.
Copy that, co-pilot.
And we see another shrimp here. There's a couple species. This bright red, I believe that's in the family Aristeidae. Absolutely beautiful image here.
Off he goes.
Okay, pilot, y'all ready for this move?
Thanks.
Is the ship stopped?
And so yeah, there was this comment earlier. Andre had pointed out that the
let's do move it due north.
That the density of animals had decreased a little bit here. The habitat also has
bridge nav
changed, so the bridge in this portion isn't as well defined.
I'd like to put in a move for 30 meters. That's 30 meters.
Our dissolved oxygen content is
2 knots.
At around 1, so it hasn't dropped that much.
Good copy.
Over the last 100 meters or so.
take a quick second to pause and get a good shot of it here. And we'll continue on.
Looks like there's another matching one just on the other side of this group.
Another matching? Oh, you think
yeah,
this is
kind of a depression here, and then one just to hop over it.
Fish. Let's get
Fish
this
left.
real quick.
Yep.
Thank you,
I
Rich.
like the fish. Okay. And get started in.
Coming in.
Yep. He's kind of far away, but
I think we have a fish. Oh, yep, you're zooming in.
Another halosaurus.
Halosaur, probably Alderman, yeah.
Yeah, it does
maybe
look
more
like.
zoom video.
Lots more.
Take it away.
Yeah,
Zoom
and that
screen.
is another alderbandia. He
Over
did
here.
zoom out, but I did get a good look at his head.
Oh, he's up near the shilling. And off he goes. I saw him run off my port side in the rail cam. He's gone. Out of reach. Sorry. Looked like another holosaur or something.
I'm not sure if Bruce has talked about this at all before, but the holosaurs are in a group.
There's that
The
other
larval
depression.
stage is actually a leptocephalus stage.
Oh, this is just a little
Yeah,
sequel.
eels like the Synaphobranchus that we've been seeing also have these leptocephalus stages, which are quite a bit different in terms of morphology from adults, from the adults, of course.
I wouldn't call this.
Excellent. Yeah, thanks for sharing that, Andrew.
No lava this old. Oh, that wasn't me.
Yeah, this is Bruce. Actually, it gets even a little bit more interesting than that. The halosaurs are in a group of fish that have very large leptocephalus larvae. And they've been found, these spiny eels, the group that the halosaurs are in, have a stage called Leptocephalus giganteus. And some of them have been close to 6 feet long, very long, slender things. But they're actually the longest fish larvae known.
So I just wanted to kind of explain some of the landscape that we've been going over the past little bit. So we've been seeing smaller little lobes of pillow lavas, but the little depression features that we've seen are actually collapses. And so what happens in these types of eruptions is that the actual pillow lava flow will inflate and subsequently drain after the volcanic eruption is no longer supplying any lava to that flow. And once they deflate, the crust on the top of the pillow lavas is hardened enough to where it will then collapse. And so those little tiny pits that we saw just a little bit ago were collapses within the pillow lava flow.
We can just get a quick look and then move on.
And just looking at the sonar, the next move we're going to be shifting a little bit northwest again.
All right.
Copy.
Yeah, very interesting that we're just seeing more and more of these massive stalked sponges.
I think we've seen a couple of these. We just want to do a
Yeah.
partial as we go by.
Sure, let's get a drive-by.
Yeah, hey Daniel, again my best guess is it's some type of sponge in the genus Saccocalyx, but I'm not at all positive.
Yeah.
It could be in a different family, possibly. I'm not sure.
Yeah, thanks, Chris. Yeah, certainly we have not seen something like this in all of our previous dives here.
Yeah, on the lower part.
Yeah.
Yeah, I'll light it up here.
So we had pictures of something like this from a hurl dive back in 2007, I believe, and I showed the pictures to Henry Reiswig up in Vancouver, who's one of the world's authorities, and his best guess was Saccocalyx on this one too.
Just see if I can get one bounce of this.
Excellent. Yeah, so just to give everyone a feel, that's probably about 1.5 meters in vertical height. So there's a big sponge.
Okay,
That's about the size of, yeah,
About done now?
a little bit smaller than I am. So that's a big sponge.
Okay. Well, I think we're gonna
Depth
shift
is 1,060 meters.
direction a bit.
So I did update our position. We covered about 350 meters
After
in
the move ends, we'll
horizontal distance.
call it in. We'll do
And yeah, we see several of these sponge stalks here to our right that
pass.
are now dead.
A lot of debris.
Yeah, and a lot of debris in here, yeah.
Copy that.
Other
And a lot of more unconsolidated boulders, looks like, and I don't know if there's instability here in the terrain.
Right, we got 2 meters
Well, we
left,
do
so
see a lot of
make it continuous.
sponge debris in some of these crevices.
Yeah, you can call it up.
Copy that.
Have them dial it in right away.
Pretty stretched out, right?
Yeah, I'm not too sure it's actually an instability as much as there might be an actual source of the rocks ahead of us, so something like a rock ledge in which they're weathering away out of.
At the end of this move, we'd like to make another one immediately. 30 meters, that's 30 meters at 015 degrees at 0.2 knots.
Looks like we just, it was a fish that was just on a quick move there, but
Good
I
copy.
see the fish. We'll get it. Sorry about that.
Oh, no worries.
Getting ready for a watch change here, but we'll get it first.
On your
Video, come on in. Get it with the lasers here. Okay, and then turn the lasers out, co-pilot.
Alright, we're going to switch up quick.
yeah, so that looks like an ophidiid.
There we go. Now he's well lit.
Thanks, Bridge.
There you go. Get some light on the subject. And then track him.
Ooh, there's a hot spot. Got a little bulbous protrusion there. underneath that
Perhaps
right eye, maybe forward
basso
of it.
gigas.
See it?
That's the closest I have here in our gallery.
Where's that?
Yeah, I see that on his nostril.
Yeah.
Now this is really interesting. This is a different type of ophidiiform than what we've seen before, I think. It doesn't look like the Bassogigas. The head's not flat enough, not depressed enough. This has a rounded head. It looks like it's got a little spine on the on the opercle. The nostrils might be informative. We'll send these images to the experts. This is good video here. So there are two families, the egg-laying Ophidiidae and the live-bearing Bathydidae. The Bathydids here in Hawaii tend to have an obvious two-part lateral line that's interrupted with an upper part in the front of the body, and then there's a gap and it goes to the lower part. I don't see that. I'm not sure what this is.
Can I get his head real quick?
Different from the things that we've seen before. There's a genus Monometopus that's been reported from one specimen from the Hawaiian Islands that I think might look like this. I don't know if Andrea has any ideas. I'll stop there.
So, Bruce, Andrea stepped out for lunch. She's meeting with Jeff Drazen for a little bit here, 30 minutes. But, yeah, it's too bad a nice shot like this when she just stepped out. But, yeah, I guess these will be good enough for you and her to take a look at later on and figure out.
I'll push up real quick.
That's great, actually. There's an interesting forked pelvic fin, too. I think that has two rays. That'll be a good character. It doesn't have that lateral line configuration of the live-bearing ones, the Bathydidae, and also the abdomen has a different shape in those. So I think this is the family Ophidiidae. But I'm not sure about the genus right now.
Excellent. Yeah, thank you, Bruce. And so as we were chasing this fish, we passed one of these sea stars. That's going to be in the genus Acinactus, and another scleractinian cup coral. But yeah, absolutely spectacular imagery of this ophidiid. There's another Anthomastus, this mushroom coral, this red coral to our right.
Yeah, it looks like once you're over the steep part ahead, you'll be okay.
Yeah, here you can really see how it's all cemented together. We're going through a quick personnel change here on our piloting team, so should be back on the move here in a second.
Okay, pilot, looks like you're just on the edge of a ridge.
And yeah, so we picked up the ridge here again, and so we'll follow that up again.
All cameras full wide.
Thank you, video.
Still got 6 meters till ship move is done.
I copy that, Nav.
The ship move was 015, is that correct?
015.
Okay. So I'm guessing based on HIPAC that either we have to keep going 015 or start turning more to the northeast.
Something in the water column there, small fish.
We're definitely on the west edge of this feature.
Top left.
And there's
According to HIPAC, I mean, it looks almost like.
And there's another fish there in the distance, and it's of course not very quantitative, but something we've. I mean, it's like 18 dives we did last year, and the last 5 dives, and this dive is areas where you start seeing more fish. You have less corals and other invertebrates, and vice versa.
Who's this little guy?
Places where you have a lot of corals, you don't have that many fish. So this is now a rat tail or macrurid.
I'm not sure we're going to get much more by following them in this orientation.
Yeah, I'm running up against the
bottom
right here.
All right, Drew, I'm going to come
Roger that.
Let him go out of frame, maybe. I'm full wide.
Well, we're looking at the underside of the fish, so I can't tell too much about it. It's interesting, though. It had dark areas on the stomach that almost looked like it could have been the areas of light organs that some of these macrurids have. It's very odd that some of these macrurids have light organs, but they're down on the stomach, and you kind of wonder what a fish would have organs on its stomach for when it lives down near the bottom. But it may be actually used for sort of communication between the species so that the individuals can signal to find mates or something like that. So it's very speculative, though.
The C-pen?
Quick snap zoom. Should be enough.
Video.
Coming in and holding.
Perfect. Yeah, that's all I wanted to see. Thank you.
All right. That's all they want, video.
Coming out. Here's your full wide.
Pushing up.
Yeah, so we just passed another C-pen, and so that's in the genus Anthoptylum. Anthoptylum.
Tiny guys are nudie. I think it's just
That's a whole separate group of octocorals, the penetulations.
Pin situated.
Copy. I'm having trouble hearing you, video.
And so there's both forms that are found in soft sediments and some that are found on hard substrates. See another one of these sacrocalyx sponges, these stalk sponges.
there's a fish passing through center, very dark.
Little guy.
Yeah.
And there's a few more of these primnoid corals.
Yeah, come on in, video.
Coming in, holding. Coming out a little bit.
Yeah, we saw a couple of crinoid and a couple of these primnoid corals, and then another fish.
Yeah, he doesn't want.
Maybe when he gets water column behind him instead of rock.
I gotta push to my port here in a second.
He heard me.
Yeah, go ahead, video.
Coming in.
I didn't get a good look, but the fish might have been one of the gonostomatids, the light fish. They're midwater fish that sometimes get near the bottom, and we've seen a few of those on these sides.
All right, come on out.
Coming out, now. Full wide.
And yeah, we see a lot of debris here, some dead sponge stalks, and some more appears to be sponge material in between the rocks.
So if you look at the Sirius cam right now, there seems to be a nice little comb jelly or jelly bobbing in and out of the view.
I'm going to bring my head back around the port.
No, I was way over there, huh?
Yeah.
Depth is 1,028 meters. And yeah, this particular feature, you don't see as many animals as we've seen before. A couple of mushroom corals and some sponges isolated.
For Sirius, there's no left-right. Yeah.
Get this thing in the water column here.
Yeah, Roger, on your command.
Yeah, go ahead.
Looks
Holding.
Your aft flat is out. I can reset.
Yeah, reset.
Like. Okay. Disabled. Rename. It's on.
Hydra Medusa.
Tighter here. Hold that.
Oh, that's way close to us. Oh, that's nice.
And
That was
down.
good. That was good.
Come on.
Thank you.
Thank you, Carl.
Yeah, one thing to point out about these Hydra Medusa is
Another ship move.
they are also in the same phylum as corals, so they're all cnidarian. So those are animals that are characterized by having stinging cells or nematocysts.
Zoom on this fish.
So I think we could do 30 meters that way.
Okay, I'm
And
saying
then-
the same thing as
Yeah,
that
go
guy.
ahead.
Yeah.
060.
Yes.
Copy that. I'm calling it in.
Okay.
And I believe that's going to be in this one subfamily Iliophinae, Synapobranchid, is in
Bridge
the
nav.
family Synapobranchidae, and
I'd like
then
to put in another ship
subfamily
move of 30
Iliophinae.
meters at 060 degrees at 0.2 knots.
Yeah, looks like that's right.
Good copy.
Thanks, Bruce. And there's a primnoid coral again. Yeah, so I was just pointing out that Hydra Medusa, the jellyfish that we saw earlier, so that those are actually in the same larger group, same phylum as you have corals. So those are animals that have stinging cells, also known as nematocysts. And then that whole phylum is also characterized by having either a polyp or a medusa life stage. So in the hydrozoa, you have both of those life forms, and in the anthozoa, you only have a polyp stage, and the anthozoa is the group that you have most of the corals that people are, most of the species of corals that people commonly refer to as corals, the gorgonians, the black corals, the scleractinians, or hard corals. But there are also stylasterid corals that are in the hydrozoa. So the term coral is actually somewhat loosely to refer to several different animals which are not necessarily very closely related,
Quick zoom on this coral.
but just have these that characteristic growth form. And so this is another bubblegum coral.
The lasers on.
Paragorgia is a genus.
No, they're not. Now they are.
Thank you. Alright, let's come on out video.
No. On the way out.
You facing the direction of move?
Yes.
Okay.
And which agrees with uphill and sonar.
Yeah, just making sure.
There's a crinoid on top of a sponge.
Let's come in on this video.
And that is a Walteria sponge, so that's the first time we've imaged that genus on today's dive. And up top of this Clyptometra is the genus of crinoid. And on that sponge you see a whole bunch of brittle stars, and that sponge in particular is interesting because sometimes you'll find shrimp growing inside of it, and in fact at one point that sponge will completely close itself and kind of form a cage trapping the animals inside of it. And again, nice little association of different organisms all coexisting. And then of course the brittle stars and that crinoid using the sponge kind of like a platform to. And there's also a squat lobster, I believe. Yep. Yeah, beautiful top-down view on the sponge.
Alright, when you're happy, Ed.
I'm definitely on the move.
Alright, coming out. I'm full wide. That's a nice float.
Coming up. Thanks, Ed.
So depth right now is 1,015 meters. Mary Wickstein from Texas A&M University just provided tentative ID for that squat lobster that was on inside this Walteria sponge.
Do a partial zoom here.
Tentative in the family Munidae.
Holding.
And that's a dead sponge stock.
This guy, come in tight on this guy.
Nudibranch?
Little sponge piece.
Sponge,
Yeah, I think that is
sponge.
exactly what it is, a little piece of sponge.
Some stuff underneath there.
There, you mean? There's a oporoid hanging out
Yeah,
back
hard to
there.
ID for that view.
Okay, I'm coming up in 75 degrees.
Oh, let me pop out.
So again, want to just welcome all of our viewers who might be watching this at one of our public viewing displays that have been set up around the state of Hawaii as well as in other places, the country, particular at the Maui Ocean Center, the Waikiki Aquarium, and the Mokupapapa Discovery Center. So you're watching live video footage that's being brought to you from the middle of the Pacific Ocean and about 1,000 miles away from Honolulu. We're diving in the northwestern Hawaiian Islands. We're on a 3-week expedition to explore the deep waters off the northwestern Hawaiian Islands, including waters both inside the Papahanaumokuakea Marine National Monument and slightly out of it. Today we're diving on the seamount about 1,250 miles away from Honolulu.
Zoom on this
And up until yesterday, we did not have maps of this area, and this is the very first time we're diving on this seamount. Yeah.
This is the same guy we've looked at before, right?
Yeah, that's correct.
Okay, looking backwards at you.
And
All right, come on out.
so, in addition to viewing this video at these aquariums and visitor centers, you can also tune in online and watch this video. We will be diving in the next couple days.
The hill is about 12 meters
but the Okeanos Explorer is actually going to be doing work here in the Pacific through September of this year, and there'll be live video footage available from the internet at oceanexplorer.noaa.gov. And so you folks can follow these expeditions in real time over the internet.
And the ship has stopped.
Yeah. Yeah, that's why I wasn't.
And yeah, this is a nice view here again of the drop and a few more animals here on this.
Yeah.
Quick zoom
This
here.
ascent.
Video.
There's another sponge, and then there's a crinoid, a feather star.
Pilot, I'm going to bring my heading to port a bit. Looks like that's the direction of uphill also.
Yeah, copy.
And that's Primnoid
Yeah, copy.
coral, along with its ophiuroid commensal. Ophiuroid or brittle star is a common name.
And coming out.
Come on out.
We're just passing 1,000 meters depth as we're going to climb up and continue ascending along this ridge. And we've seen that the ridge is narrower in some places and then widens out. And these narrower features are the places where we've been seeing more animals. And that's a common theme in that.
Quick zoom down here. Fish shrimp. Yeah. See if we can come in tight as I come by.
And this is another one of these long-legged shrimp, also fairly common, and this is in the genus Nematocrinus.
Alright, when you're happy. How am I doing, co-pilot?
I'm pretty much stopped, but I'm looking backwards at you.
Alright, copy.
Looks like 030 is uphill. That's where I am.
I'm working the center up on you.
So, yeah, we spent about 3 hours, just over 3 hours on this dive. So we landed about 3 hours earlier at close to 1,300 meters. We're now at 1,000 meters. Over the course of the dive, we did collect a rock sample. And so at the beginning parts of the dive, we did see some nice moderate to higher densities of primnoid corals. And on this part of the ridge, the density is now decreased, but we're seeing more and more of these very enormous stalked sponges, tentatively ID'd as glass sponges in the genus Sacocalix. And some of these are 1.5 meters in height, so the size of a small adult, which is quite remarkable. And then also want to give our shore-based scientists a heads up. So we do have about 5 hours remaining on the dive.
Let's do a quick zoom here.
About an hour from now also we will, oh that's interesting. Oh, so that's a Chrysogorgid.
Do you have any more, Ed?
Yeah.
Yeah.
That's it.
Okay. I'm going to move in on it.
Someone just call in?
Hi, Daniel. It's Scott Friend from Louisiana.
Hey, Scott.
Getting set up. Nothing bad at this point.
Excellent, thank you.
I wonder if I could go from that Oakroy.
But I'll be standing by if you need me.
It's just
Copy that. Thanks, Scott. So yeah, I was just saying that they want to give folks a heads up that about an hour from now we're going to be experiencing one of these sun fates, this very unusual phenomenon that we're going to be experiencing every day, where we're going to lose connectivity, so the phone and video might cut out. So that's happening a little less than an hour, so just to give you a heads up. And here's another type of Hormatia anemone. So this is another Venus flytrap anemone. This is in a different genus and a different family, for that matter, than one that we saw earlier.
Hey, come on in. I'm going to move in on it.
So this is going to be in the genus Philiactis and in the family Hormathiidae.
Now, while we're waiting, maybe we could zoom out on HiPAC and get an idea of how far we've come and how far we have to go to the top.
Still.
To waypoint 2?
Yep.
And how far have we come since...
I do not know. Let me check right now.
Okay.
I have more zoom when we get our fixed focal length. Sorry, fixed distance.
Looks like 372 meters since
Well,
waypoint 1.
Oh, I see. The rock was on the bottom, though. That must be where we hit the bottom.
Yeah, so let's see exactly what we got in there.
Okay.
So, Daniel, I'll just say a few words about the anemone. Not my specialty, but I know something about these things. The Hormathiids are sort of diagnosed in part by the very textured stalk. It's a very sort of chitinous type of stalk. And then right below those tentacles, there's sort of a clear zone of tissue before the tentacles come out. So it's fairly easy to spot these things from a distance if you know what to look for.
Yeah, thank you, Chris. Yeah, and you're definitely, the stalk is very distinctive. That's another sea pen there, a rock pen. Seen a few of these. It's going to be in a genus Anthoptylum.
3.5 hours ago.
Yeah, so I was talking about this very unusual phenomena, the sun fate, that will happen in a little less than an hour, and that's based on our position and the position to the satellites. And it's been quite interesting that we're actually, because of where we are, seeing a few of these interesting phenomena that last evening, in fact, we had a solar eclipse that was only visible for about a band about 100 miles in a very narrow stretch up at these latitudes, and we all were able to see that, so that was quite remarkable. So now, yeah, we've passed 1,000 meters. And yeah, I agree with you, Chris, that stocked sponge it will be a good candidate for collection. It does seem to be fairly common, and but we'll try to cover some ground before we maybe when we're at a good stopping point. It seems to be so common. In front of us is another Metallogorgia. And that's a octocoral and a Gorgonian octocoral. The Gorgonians, they have a skeleton axis, so which differentiates them from like the true soft corals, which don't have such an internal skeleton. They still have calcite or skeletal elements that are fused, but don't have this rigid axis running around the center of the animal. And so the mushroom corals, those are the alcyonaceans, or what people typically refer to as the true soft corals. And that red coral there towards the top is a representative of that. And then I think the coral to our left, that's the first time we've seen that. Maybe quick just snap zoom to so yeah, I agree with you, Chris, that looks like Chrysogorgia geniculata.
Coming in. Holding.
Yeah, so Daniel, just a minute ago we passed by a smaller stock sponge, and that most definitely was the Saccocalyx, and then a little further on was this larger one, and so it brings up the possibility that we're just looking a small one that looks kind of classic for that form, and then as they get older, they get all kind of twisted like those bigger ones. Mm-hmm. So that's a real possibility of what's going on there, and it may just be something that we're fully aware of already.
Yeah. No, and you had brought that up earlier, and yeah, it would make sense since that's the main sponge that we've seen, and we're seeing both the small and the big ones in this kind of environment. And so do you think we should is it still worthwhile to collect one of these, or are you leaning towards they might be the same thing?
I would say wait till you make some more distance, and you're probably going to get into more animals as you get closer to the top, and might have a better candidates there. And then if you're desperate toward the end of the dive and you come up against, or you come by one of those big ones that might be worth collecting.
Yeah, I totally agree with you, Chris, and yeah, glad you say that. Yeah, we do want to make it to the top.
Coming out.
We'll make that a top priority here for this dive. We do have
I think we're good
nice
for the
bottom,
next
another nice bottom time remaining, but yeah, it's towards that top of the ridge where we're hoping to find the biggest
030.
increase in density.
Copy.
That's the way I'm faced now.
Copy that. 30 meters at 030. Bridge nav. We'd like to put in a ship move of 30 meters at 030 degrees at point.
It's a pretty weird little coral on the left there.
Yeah, maybe a quick snap zoom.
Good copy.
This guy on the left?
Yep.
Copy. All right, come on in, Ed.
Coming in. Partial.
So it looks like some type of primnoid. It may be just kind of a deformed version of a Norella or a Calyptrophora wyveli or something. I'm not sure.
Yeah. Yeah, it's definitely a primnoid, but...
Thank you, Bridge.
Polyps are downward facing.
co-pilot. Just so you know, some of those moves were really quick when we weren't on. And they covered a bit of ground while we were.
Coming out just a little bit. Holding there.
We'll try to keep moving then. The pace.
So just to give folks a heads up, so weather permitting, we are going to be diving tomorrow and the day after. And then by Friday evening, we'll have to start steaming down to Kwajalein. So this leg will end in Kwajalein on the 18th. After that, the Okeanos Explorer is going to head up to Wake Atoll and do a mapping-only cruise, and then head down to the Marianas Trench Marine National Monument for more remotely operated vehicles. And then moving its way back to Hawaii, stopping in the Pacific Remote Islands Marine National Monument. So yeah, for the next 6 months, the Okeanos is going to be doing work up here in the U.S. Pacific. And this is year 2 of a 3-year effort to study and survey some of these unexplored deep water environments in and around the marine national monuments and sanctuaries of the U.S. Pacific. So yeah, some of the most significant treasures in terms of natural resources of the United States are found in these remote places in the Pacific, and a lot of those treasures are found in very deep water. So most of these marine protected areas are very large, and the vast majority of their waters are contained in very deep waters, and of which we know very little about. And thanks to this campaign by the Okeanos Explorer, we'll have a much better handle of what is found in what is arguably the largest portion of these monuments and their biggest contribution in terms of space. So yeah, that's another one of these Astenactus. We had imaged one earlier.
Oh, okay. Alright, then we'll just take a quick shot.
So that's it's a sea star in the genus Astenactus.
Alright, coming out.
Sirius has started moving. You're in a good position now.
Copy.
That is full wide.
Dan, this is Bruce. Do you have an idea of just the general area where the dive tomorrow will be?
We'll discuss it after, but there's a couple seamounts that are in our vicinity that are just outside the monument as well. The transit consideration, depending on when we're ready to transit, is going to play into that. But yeah, there's 2 additional seamounts that are right outside the boundaries here that have some interesting topography.
Coming in.
So Daniel, is that coral in front of us in the Thanopathes? It almost looks like that twisted one. No, I guess not. Sorry.
I did not see it.
No, that looks like it's primnoid. Sorry, miss ID from a distance.
Yeah, so this is definitely a primnoid, what we're looking at. But yeah, it's interesting. Today, on today's dive, we have seen 0 black corals. It's been primnoid corals, a few bubblegum corals and pink corals,
Small objects in it.
very few bamboo corals, a couple unbranched isidids, but yeah, predominantly this community is dominated by primnoid corals and by this sponge right here, the Sacocalix, which we think might be small and large individuals, and they're
just missing some branches.
yeah.
The move is just about complete. Do we want to line up another one afterwards?
Coming out.
Yeah, I'm looking down 70 degrees now, so I'll have to...
Yeah, this is Bruce again.
Sure the
So
ROV
the
gets set
dive
ahead.
tomorrow definitely will not be as far north as Helsley Seamount. Is that correct?
Yeah, that's outside of our reach. We are too far to the southwest to reach that on either of these 2 days. Yeah, we were trying our hardest here during to get to the northern portion where most of our targets were for this
down at you 80 degrees.
for this expedition, but just the weather did not cooperate. We'd had multiple storm systems just hanging out right above Midway, so we had to stay
a bit, yeah,
further south than we hoped to be.
Camera over there looking at it.
Yeah.
Yeah, I feel
Yep,
like
I understand. We got the strong winds last night that I think you had up north, and I think they're still going on now. So I figure that you folks must have been really uncomfortable up there with the ship rolling around. So.
Yeah, you can absolutely, say that we've had some rough seas, and so this is the first time we've seen this animal right here. This is the one below it that was Iridogorgia magnispiralis, but I believe that's a young Metallogorgia melanocristus, and Les Weiling had pointed this out a couple days ago. So yeah, this one right here is Iridogorgia magnispiralis. Individuals of this. But going back to the coral above it, the young Metallogorgia, so Les Watling had pointed out these young ones, they have multiple plumes, and then later they lose the lower plumes and just left with the top one. So yeah, we've had unfortunately some very rough seas on this expedition, and that's on par for this part of the ocean and this part of the year.
There we go.
For those folks who are not from Hawaii and the winter months, we have very, very large waves, and in fact this year was unusual even for this time of the year. There's this big surf wave competition, the Eddie Cow, that happens when there's large waves and that they're consistently large, so they have to be consistently bigger than 30 foot on average, and that's a rare thing. This competition has happened a total of 9 times since it was first started in the mid-1980s, and the last time we had those kind of conditions was 6 years ago. So yeah, this has been a tough winter, and yeah, we had some very unpleasant seas here.
So actually, it feels like I have a current from the coming out of the west now.
Copy that. Thank you.
I'm not 100% sure, but that's what I'm feeling.
Nav, what's the general uphill direction on head back now?
And I'm just looking over here our dissolved oxygen sensor, and it was the first time we're reaching levels below 1 milligram per liter.
Locally, it's about 35.
So where we landed at 1,300 meters, we had values around 1.5, and then it gradually was decreasing. And now we're seeing the first time numbers below 1.
Up onto the ridgeline, I think.
And yeah, so throughout this dive, we should be transversing this oxygen minimum layer, and then
maybe 010.
Probably around 200 to 300 meters shallower should start coming back up those oxygen levels, and we'll see how the communities change in tandem with these changes in environmental variables.
Yeah, I mean.
Yeah, locally there's a little bump here. Could move that way.
Quick zoom here, video.
Coming in.
Sure they've seen this, but.
Yeah, that's another Narella, and that looks very much like the dichotoma.
Alright, coming up.
And coming out.
Yeah, so these primnoid corals, Narella,
Thinking maybe
definitely the most dominant ones here.
I think we're kind of zigzagging.
Yeah, I think this is the top right here.
Yeah, so.
Yeah, if you look, it just drops off. So.
If we did like 010, I would approach that to center over it.
Sure.
Fish upper right.
Okay. Yeah.
Okay. So 010 or 000?
I think 010.
Copy.
We don't want to keep overshooting it, zigzagging.
We'll take a...
On the rat tail?
Yep.
Roger. Holding.
Bridge nav.
And that looks like Coryphonoides.
We'd like to put in a ship move of 30 meters at 010 degrees at 0.2 knots. Good copy.
But he does not want to show.
Yeah, but it looks distinctive enough that I think it might be Coryphonoides longiceros, except the pelvic fin rays aren't long as they are in that species. So could be a different Coryphonoides species than what we've seen before.
Bank turn.
Excellent. Thank you, Bruce.
And now we've got excellent video. Now we can see more. It turned very nicely. So got scale pattern on the head. We have a lateral view, so we can send some of these images to Tomio Iwamoto at California Academy of Sciences, who is the expert on this group of fish, and he's been very helpful with identification.
Excellent, yeah, and some very exciting things with all these new IDs. Bruce has shared with us earlier that we've with these very few dives last year, 18 added. 25% of the records were new for Hawaii. So with just a little bit of a few of these dives, we're really expanding the known biodiversity from this area. And it shouldn't really surprise us, since we are looking at places where no one has looked before, and so we are finding new things.
Back up.
You see the lasers? Oh, there they are. Bam.
So yeah, those are, again, that's a rat tail fish. The scientific name for that family is the Macuridae, and that's derived from it's basically macros and uridae, long tail.
So yeah,
Just
local ridge is just to your port.
Yeah.
And these are some of the most abundant and common fishes in the deep sea.
I'm facing the direction
Another
I'll be moving, so hopefully
common name for that group is the grenadiers.
Yeah, and I started moving a little bit ago.
Excellent. Thank you.
Yeah, I just want to also,
Just further away, the amount
again,
of snow
thanks
is
to all of our
increased.
shore-based scientists who have been absolutely wonderful and helping us out today. We again have a nice group joining us. We got Andrea Quattrini from USGS following us from the Exploration Command Center at the University of Hawaii. Asako Matsumoto from Japan. Brendan Rourke from Texas A&M. Brian Boston from the University of Hawaii. Bruce Mundy from NOAA's Pacific Island Fisheries Science Center. Chris Kelly from the University of Hawaii. Diva Amon also from the University of Hawaii. Les Watling, also University of Hawaii. Mary Wixton from Texas A&M. Mike Vecchione from the Smithsonian Institution, National Museum of Natural History. Nicole Morgan from Florida State University. Scott Framps from University of Louisiana, Lafayette. So nice assemblage of some of the world experts in a lot of different groups.